Hello, my name is Kristi Anderson from Group 5A. I will be reviewing the important concepts from Chapter 38, Plant Transport. First, let's begin with a general overview of plant transport. In most plants, the root system absorbs water and dissolved minerals from soil. The shoot system takes up carbon dioxide entering through plants via pores on the surface of leaves, called stomata, in addition to other structures that are above ground. Water, minerals, and carbon dioxide are used by photosynthetic cells to produce sugars and organic compounds for growth and reproduction. Non-photosynthetic cells, such as cells of plant roots and flowers, depend on the nutrients produced by green tissues. Water and minerals are transported upward from roots and shoots. Nutrients from photosynthetic cells are also transported to non-photosynthetic portions of the plant. Long-distance transport occurs in a continuous system of conducting tissues. Remembering the function of xylem and phloem, these conducting tissues are key to plants living in terrestrial habitats, with xylem transporting water and dissolved minerals and phloem transporting organic substances in watery sap. Moving on to uptake and movement of materials at the cellular level. Plants use both active and passive transport to import and export materials. Passive transport is the movement of materials down a concentration gradient, and it does not require ATP. There are two types of passive transport. Passive diffusion, which is the movement of molecules through a phospholipid bilayer down a concentration gradient. And there's facilitated diffusion, which is the transport of molecules down a concentration gradient using transport proteins. Channels and transporters are two main types of membrane transport proteins. Osmosis is the diffusion of water across a selectively permeable membrane in response to differences in solute concentrations. The movement of water is from a low solute concentration, as in soil, to a root cell, which has a higher solute concentration. Osmosis is essential for plant cells for cell expansion, structural support, and to perform photosynthesis. Aquaporins are a type of protein channel that increase the rate of water flow into expanding plant cells and vacuoles. Active transport is powered by ATP hydrolysis. Membrane transport proteins use energy to move substances against their concentration gradient. The hydrogen ATPase proton pump is an example of active transport. This proton pump is found in the plasma membrane of plant cells, which uses ATP to pump hydrogen ions against a gradient, as illustrated in Figure 38.2b. Turgor pressure is the hydrostatic pressure that increases as water enters plant cells. The cell wall of plants restrict the extent of how much the cell can swell. The water content of plant cells depends on turgor pressure as well as solute concentration. A plant cell full of water causing the plasma membrane to press up against the cell wall is considered turgid. Therefore, the more water a non-turgid cell absorbs, the more rigid it will become as water exerts pressure on the cell wall. Figure 38.4a shows an example of a turgid cell. The plasma membrane presses tightly against the cell wall. Figure 38.4b shows plasmolysis, which is a condition where the plasma membrane no longer presses up against the cell wall. The cell has lost water by osmosis, and turgor pressure has been lost, resulting in a plasmolyzed cell. When the concentration of solutes is the same outside and inside a plant cell, it will be flaccid, as shown in Figure 38.4c. Solute concentration and turgor of a plant cell influence a property known as water potential, or the potential energy of water. Potential energy is the stored energy of a system with the capacity to do work. Water moves as a result of gravity, from a region of higher water potential to a region of lower water potential. Pressure and certain solute concentrations also influence water potential. When studying plants, water potential can be applied to understand the movement of water into and out of cells and to understand the water status of an entire plant. The water potential equation is used to predict the direction of cellular water movement. Using information about the solute concentration inside and outside plant cells, in addition to the pressure exerted between the cell and plasma membrane, water potential can be calculated. Relative water content is used to measure the water content of a plant, taking into consideration the water potential of all cells within a plant, measuring the overall turgidity of the cell. The relative water content can also be used to measure a wilted plant's ability to recover. Measurements less than 50% typically predict death. More importantly, these measurements have been useful in ecological studies. Plants have adapted to cope with extreme environments that cause low water content. Osmotic adjustment is the increase of solute concentrations of cell cytosol. Plants in arid lands utilize salts to stabilize cell membranes to prevent damage during plasmolysis. Plants are also able to adjust to saline environments by acquiring adaptations such as an excretion of salts from root surfaces. Tissue-level transport occurs in three forms. Transmembrane transport, which involves the export of a material from one cell, followed by import of the same substance by an adjacent cell. Second is symplastic transport, which is the movement of a substance from the cytosol of one cell to the cytosol of an adjacent cell through membrane-lined channels called plasmodesmata. Symplastic transport has a potential to move molecules widely among the cells and tissues of the plant body. Apoplastic transport is the movement of solutes along cell walls and the spaces between cells. Nutrients usually move plant tissues for short distances by apoplastic transport. Symplastic and apoplastic transport are important in mineral nutrient transport through the outer tissues of roots. In the roots of plant cells, the endodermis is a thin cylinder of tissue composed of close-fitting cells and specialized cell walls, forming a barrier between the cortex and the central core of the vascular tissue. Casparian strips, as illustrated in figure 38.8, located in the root endodermal cell walls, prevent apoplastic transfer through the endodermal cell walls into the root vascular tissue, which prevent harmful solutes such as metal ions from moving through the apoplastic vascular tissue and being transported to the shoot. Endodermal plasma membranes also possess specific channels and transporters for essential mineral nutrients. The root endodermis functions as a molecular filter that allows the passage of beneficial solutes. Moving forward to long-distance transport in plants. Plants possess an extensive branch long-distance vascular system composed of xylem and phloem. Bulk flow is the mass movement of liquid caused by pressure, gravity, or both. It occurs when molecules of liquid all move together from one place to another as a result of differences in pressure. It also allows mineral ions to move through the soil to plant roots. Specialized tissues foster bulk flow and aid plants in adapting to water stress. Evaporation of water at plant surfaces, cohesion of water molecules, and adhesion of water to the walls of cells are important and influence long-distance water transport. Xylem plays an important role in transport. Xylem contains several types of specialized cells, such as tracheids and vessel elements, together known as tracheary elements, which contribute to structural support of the plant body and aid in transport. Figure 38.11 shows tracheate cells in xylem tissues. These cells are long and tubular with slanted end walls. Water and ions move through the pits. Vessel elements have a wide diameter with several pits and end-all perforations. This structure allows water to flow faster from one vessel element to another. Cohesion, tension theory, and transpiration. Transpiration is capable of pulling water by bulk flow. Plants expend no energy transporting water and minerals by transpiration. The sun indirectly powers this process by generating a water pressure difference between the moist soil and dry air. This process moves a huge amount of water from the soil to the atmosphere. It also plays an important role in the bulk flow through xylem. The cohesion tension theory explains long-distance water transport as the combined effects of cohesive forces of water and evaporative tension. Phloem plays an essential role in long-distance transport of organic molecules and minerals. Sieve tube elements are arranged end-to-end similar to vessel elements of xylem. Together with companion cells, they form a transport system. The perforated end walls of mature sieve tube elements form sieve plates, and there are perforations known as sieve plate pores. Phloem sap passes through these plates from one sieve tube element to another. They lack a nucleus and are dependent on companion cells for mRNA and proteins. Masses of protein accumulation occur when damage is done to the structure, preventing loss of phloem sap, shown in figure 38.22 on the right. Companion cells also play an essential role in moving sugars into sieve tube elements for transport, a process known as phloem loading. The final topic for Chapter 38 is the pressure flow hypothesis. Phloem transport is driven by differences in turgor pressure that occur between cells of a sugar source, where sugar is produced, and those of a sugar sink, where sugar is consumed. Examples of sugar sinks include roots and developing leaves and seeds. Translocation is a process where the phloem transports substances from source to sink, which can be horizontal or vertical. Sieve tube elements near source tissue typically have higher solute concentrations than surrounding tissues. Therefore, water rushes in, building turgor pressure. Sieve tube elements near sink tissues have lower solute concentrations, which drive the bulk flow of phloem sap from source to sink tissues. Figure 38.24 illustrates the pressure flow hypothesis. This concludes a discussion of key concepts for Chapter 38, Plant Transport. Thank you.